So today, we're going to talk about inflammation and aging. It's a very hot topic. Judy Campisi is here. Judy's an internationally recognized expert in the field. She's a pioneer. Just happens to be on the Buck faculty, so believe me, you're in for a real treat today. Some housekeeping issues before I get started. I am required by law to point out the emergency exits in the back of the room. I'm also asking you to turn your cell phones either off or put them on airplane mode because the wireless signals sometimes get in the way of good audio here in the room. I want to say a big hello. There are folks who are in our overflow room, so I want to say hi to you. You are definitely a part of this event. Hopefully you're enjoying a cup of coffee and a muffin because that's one of the advantages of not being in the Drexler is that you can have treats. You will be included in the interactive part of the event, so pay attention because we'll be getting back to you. I also have some thank yous. I want to thank our staff and our volunteers. We really appreciate time out of their everyday activities to make this event a success. And I want to give a special shout out to our IT team. Those folks went above and beyond the call of duty to get our overflow room working today, so folks can enjoy the event from there. So some of them are in the back of the room. Thank you guys, really appreciate it. Yay for IT. I also want to thank Novato Public TV. They're here today videotaping the event. It will be online, so if there's anything that you want to review from today's event, you'll be able to do that online. And speaking of thanks, this event would not be happening without the generosity of the Levy family. Mrs. Levy has sponsored an event here at the Buck for 12 years, and we're so grateful for her support. Mrs. Levy, why don't you stand up? I was gonna have you stand a little bit later, but this is Mrs. Levy. Thanks again for your support. So, these events are done in honor and remembrance of Dr. S. William Levy. He was a renowned dermatologist in both San Francisco and Marin County. He was also a clinical professor at UCSF. He was a published author and an international lecturer. As you can see from this slide, he also had a really great sense of humor. So I love this photo. He was a visionary. He became involved in the Buck Institute in 1993. That's 6 years before this building was even opened. So he was very involved in helping set the vision for the Buck, and he was one of our earliest supporters. Mrs. Levy remains a key supporter of our events, and I also want to call out Dr. Levy's brother Mike is here. Mike, just wave your hand. Mike is 102 years old. I know, a great example for all of us, and he told me beforehand that he celebrated his last birthday at the ballpark where he had 35,000 people sing happy birthday to him. Not a bad way to celebrate your birthday. So, our hope is that you are all inspired by Mrs. Levy's generosity and that you support our research here at the Buck. From the turnout and from those of you who I know personally, I know that you're really eager to get good, solid information about how to age well, live longer, better. This is the place to do it. It's based on research, it's based on science, so that's why we really do need your support. After the event today, there's a table outside. There are donation forms. We're also now in the new century, we're able to take credit cards. So if you want to pay with a credit card today, we're happy to do that. Chloe's in the back of the room, raise your hand. She's the maestro of the credit card swiper, so track her down if you so wish at the end of the event. So now it is my pleasure to introduce our CEO, Eric Verdin. Eric is the guy who has given this place great energy and is directing us into the future. And he's going to give a bit of an update on some of the activities here at the Buck. He's also going to introduce Judy.
Thank you, Chris, and welcome all of you to the Buck. So if you've been here before, thank you for sticking with us. I think we are in a really exciting period of the life of the Institute. If it's your first time, I hope this will be inspiring enough and exciting enough for you to come back and join our community. As I've mentioned a number of times, I think we really believe in the idea of an institute being anchored in its community, and that is the whole Bay Area, essentially. So it's, who's here from Marin? How about Napa? We have to work on Napa. No, Sonoma. Fantastic. San Francisco. East Bay. All right, okay, so for the rest of our communication team now, we know, we've taken note, we know how we have to, who we have to call for. Anyway, we really want to be the institute of the whole Bay Area, and eventually, the country and the world. The work at the Buck Institute is really pioneering. We were the first one as a free research institute doing aging, and we were doing it before it became popular. And believe it or not, it is becoming incredibly popular. Judy and I were just at a meeting in Europe, actually a day ago, and a meeting on aging, the field of aging is just exploding in terms of discoveries, a lot of excitement, a lot of interest from potential commercial partners to actually bring all of these discoveries to the clinic. So if you're joining the aging field, you're doing it at an amazing time, which I think will change humanity in the way we really deliver healthcare in the future. So as part of my job, I also want to bring you up to date on some of the key developments that have happened. I'm coming to my 2-year anniversary here at the Buck. It started on November 2016. And I think proudly can say that we've really made great progress in terms of changing the narrative, in terms of really looking at the Institute as a place that's going to grow in the future. And part of our growth is to recruit new talent. And so we have 3 new recruitments actually that just happened in the last few months, and I want to introduce them very briefly because you probably will hear from them at future seminars. Our recruit here is Tara Tracy, she's just joined us as an assistant professor. She works on Alzheimer's disease, and she works on a unique protein called tau, which is emerging as really one of the key new players. As you've heard, there's been a lot of disappointing news on Alzheimer's treatment. It was mostly based around studying one protein. Well, the field is now moving to the next one, and it turns out that Tara has really been one of the young people pushing this story on tau. So we're quite excited to have her. She has a PhD from Berkeley. She was a postdoc at the Gladstone Institute in San Francisco and at UCSF. Could not be more happy to have her. So she might be actually here afterwards. So if you see her, say hello and welcome her. A new member of our faculty is also Birgit Schilling, assistant professor. Birgit has been here at the Buck for 18 years. She's been running a core. A core facility is actually a set of technologies that everyone at the Institute is using. Her specialization is called mass spectrometry. It is one of the areas of biology that is currently exploding. And Judy has shown great leadership at the Institute, not only helping everybody else, but also developing her own research project. And so she was just made a full member of our faculty. She's originally from Germany, and she was also a postdoctoral fellow at UCSF. And again, if you see her, say hi. We're really excited to have her. I also want to say one word about a really exciting new opportunity here at the Buck. We received a large gift from a family that lives here in the Bay Area. And this family, actually, they're linked to Sergey Brin, the founder of Google. And they are quite excited about supporting a new program here at the Buck, which is what we call the Center for Female Reproductive Longevity and Equality. And this is a completely new area of focus. It's relatively been unstudied, not only here at the Buck, but everywhere else in the world. And it's a reflection of the fact that close to 10% of women suffer from infertility. And so this is really, and it turns out that the mechanisms that drive the aging process are at play. So the prediction is that by studying infertility in the context of knowing everything we know about aging, we're going to be able to address a really significant need in our society, especially as women entering the workforce are delaying childbearing. This problem of infertility is getting more and more significant. And the solution today is to freeze eggs in young women, and we think we can do better. And because also it turns out that early infertility in women is a predictor of a shortened lifespan as well. So the two are tied very closely together. So we're quite excited about this new program. We are basically will be focused on ovarian aging. We're exploring ways to use the center for community engagement with women's group. We want to take the leadership in this issue. And we're currently recruiting 2 new faculty members to begin staffing this center. So stay tuned. And if you read in some newspapers that we are planning to make 70-year-old women have children, just tell them that you know better. This has nothing to do with this. And we'd just be happy for all 40-year-old women to be able to have children. That's actually really something that we are focusing on. Now, on to the topic of the day, and Judy just closed her eyes, and because she knows I'm about to embarrass her. So, but this, I'm sorry, Judy, I have to do it. So, we selected Judy to give this lecture and to represent the BUC today because she really epitomizes everything that I think not only me, but our whole faculty, our community, views as science at its very best. And so I want to make sure also you understand we're not deluding ourselves thinking that we're great, because Judy was just elected to the National Academy of Science this year. So this is, if you're not aware of what the Academy of Science is, it is the ultimate honor that you can get as a scientist. The whole academy for the whole country has less than 2,000 members. Every year, 80 scientists get elected for all sciences. It's not just biology. This is physics, mathematics, everything. And so the fact that Judy was selected in this incredible group is a validation of what the BUC is doing. Just to give you an idea, all of UCSF also had 1 member, and UCSF is a 25,000 employee organization. We're a little bit smaller, but we're punching way above our weights. And so having an academy member here is really incredibly important, not only for the recognition, but also for the significance that it has for aging. So aging is a brand new field of research, about 25 years old, at least molecular aging, the way we do it. And the fact that now we have academy members is really a recognition from the establishment that the science is important, and we are key players in it. Judy, in some way, in many ways, actually, the reason we really wanted you to hear about her is that she is what I call a scientist, like a doctor's doctor. Her work has just been incredibly pioneering. We define pioneering by the idea of being willing to study a problem before most people think there is even a problem, and or to identify novel treatments before people think that this could actually be done. So Judy has really been a key player in the field of senescence, and the field of senescence today has just exploded, not only in terms of our understanding of its role in the aging process, but also in its translation. And Judy's been associated with a company that is now putting drugs into the clinic for the first time, and we think that company, I suspect you'll be discussing this in much detail. That company called Unity is really one of the first ones to enter the clinic from the standpoint of the aging field. And we predict, not only us, that as they become more and more successful, the whole field of biomedical research is going to say, well, there are incredibly important opportunities to be made in this research. So, Judy, I'm delighted to have you today representing us, and thank you very much.
So while Judy takes her seat, I want to acknowledge her courage for letting me control the clicker. It's really a challenge for a scientist to give up the clicker, so I want to say that.
It is.
And also, here's how it's going to work. If you have questions for Judy, I think you all got index cards. If you need an index card, raise your hand. We'll get one for you. At some point, I'll say it's time, and I'm going to have volunteers collect those cards. Also from the overflow room, so if you have questions, please ask them, and then we'll go on from there. So, let's see if this clicker works. Yeah.
Yes.
So, the first question, Judy, is about cellular senescence and what it is, and I want to tell folks, I put this slide up not so much to freak you out because it's so complex, but to give you a sense of how complex the topic is. And at this point, I just want you to pay most attention to the teeter-totter part of the slide and that happy face on the left and the grumpy face on the right. So, Judy, give me your overview of what is cellular senescence?
Well, it makes people happy and it makes them grumpy.
So what is it?
Okay, so the cells in your body are constantly under threat of stress. And that stress can lead to two things. The cells can fail to function, or worse, they can begin to proliferate inappropriately and give rise to a tumor. So cellular senescence is a way for a cell to sense whether it's stressed or not, and then it opts to stop proliferating forever. So no cancer, so it's a very potent anti-cancer mechanism. At the same time, it starts telling its neighbors, hey, something's wrong. And we call that the secretory phenotype, the secretion of molecules that can help that cell communicate with its neighbors. Now, sometimes that's good. For example, if you cut yourself, you know how it turns bright red? That's called acute inflammation. It's absolutely essential. You'll never heal that wound if you don't have that reaction. And it starts with senescent cells communicating something's wrong. So that's good. The bad thing is these cells slowly accumulate with age. And they're still secreting these molecules that's causing inflammation. That's chronic inflammation. And that's bad. That underlies every disease that's associated with aging, has some component that's chronic inflammation. So that's the happy face and the grumpy face.
So here's a slide that gives a sense of what happens inside the cell. So you've already touched on it, but what is in that SASP that you talk about, the things that come out of the senescent cells?
So the stuff that comes out of the senescent cells are lots and lots of molecules. Actually, we first discovered this in 2008 when technology was, well, you know, in science things move so fast. So this was like the Neolith with regard to technology. And we identified 40 different molecules that the cells were secreting. Since that time, actually in large part due to our collaboration with Birgit, who runs this technique called mass spectrometry, we've increased that by more than tenfold. So we now know there are hundreds of molecules that the cells are secreting. And some of them are designed to attract the immune system and cause this chronic inflammation, but some of them are designed to help a tissue undergo repair and remodeling, which is something that we all need and slows down with aging. So again, it's this double-edged sword. It's a balancing act. And I hope we'll talk later about how we separate the good from the bad, because that will be critical.
So here's this the next slide. This one might be easier for you to read, but I actually think it's a little scarier than the other ones. It's kind of a list of the diseases that senescence is associated with. So why don't you talk about some of those, the major diseases there, Judy?
So in the last 5 years, we have now made, so this is mostly in mouse models, but we do have some data in human. We've made mouse models where, due to a transgene, so a totally artificial gene that we insert into the genome of the mouse, and we make it possible to selectively kill only senescent cells in that animal. It's the first time this was ever done. And using that mouse model, we were able to show that it was very important for, senescence was very important for suppressing cancer, and we were also able to show that it was very important for wound healing. Now, the beauty of science is none of us work alone. We have collaborators all over the world, and we've sent them our mice, and those collaborators are working on these diseases. So everything from Parkinson's disease, which was a collaboration here at the Buck with Julie Anderson's lab, to cancer metastasis and the side effects of chemotherapy and antiretroviral therapies, all of these are driven, all of these drive cells into senescence. And with the mouse model, we're able to the first time to show that senescence is causal. And so this has revolutionized the field. When I tell you that there are bad things that senescence does, this is the list. So everything in blue is published, and everything in black is still ongoing, but I've seen most of the data, and it's pretty impressive. So for the first time, we now have a basic cellular process that was evolved for the good purpose of preventing cancer. And now we're showing that it's driving lots of age-related diseases, including, very ironically, late-life cancer.
So how does it drive late-life cancer?
By virtue of what the cells are secreting. So let me explain a little bit about the evolution of aging. So imagine we're living in an era where there are no cell phones. There are no automobiles. I know it's hard to believe, right? When did that happen? Way back, thousands of years ago, lifespans were very short for humans. And they were short not so much because of the diseases we recognize as aging, none of the diseases up here, but because of the fact that people died of predation, they died of infection, they died of starvation, they died of exposure to the elements. And so that was called a high hazard environment. It's the environment in which our species evolved. Nobody died of old age. Nobody died of these diseases. They were all dead by the age of 40 or 50 at the most. So evolution evolved ways to protect people from bad things happening inside themselves, only for about 40 years. And everything that happens after the age of 40, we say have escaped the force of natural selection, because evolution couldn't act because everybody was dead. So now, we, humans, we've done this. We've changed our environment. We have, of course, this is for the developed world, but we have control over infection. We have control over our food supply. We have control over our climate. And we're living much, much longer. But evolution hasn't had time to fix the problems we're now facing because it couldn't act when we were all dead.
Perfect lead into this next slide. If I can bring it up. There we go. So, Unity Biotechnology. So, we might be able to do something about this thing called senescence after age 40.
Yes, that's right. Yeah, so I should point out, Unity started here at the Buck. It was incubated here at the Buck Institute when really it was just a gleam in a very talented person who was working in a venture capital firm's eye. And when he approached me and my colleagues at the Mayo Clinic and said, let's start a company to develop drugs to kill senescent cells the way your transgenic mice can do it, I told him, no way. I can barely balance my checkbook. I can't run a company. I don't do that. But he persisted, and we did the science, and he did the development of the company. So the company now has over 100 employees. It's gone public. It's down in Brisbane, in South San Francisco. And it's
phase 1 clinical trial.
Exactly. It's just starting its first trial of a drug that was identified by our labs that's going into people. So those of you who are aware of drug development, you probably know what phase 1 means, but for those of you who don't, if you have a brand new drug and you want the FDA to approve it for a disease, the first thing you have to do is show that it does no harm. So that's called phase 1. And in that case, what you do is you give the drug to a bunch of volunteers and you cross your fingers and hope nobody dies. So nobody has died. But they're very smart about the way they're doing it because the FDA does not recognize aging as a disease. So what the FDA wants to know is which age-related disease are you going to go after? And they decided to start with osteoarthritis and glaucoma. Why? They are prominent age-related diseases, but also the administration of a drug can be local. So they're doing local injections into osteoarthritic joints, and so far nobody's leg has fallen off and nobody has died. So that's good.
That's great. And then assuming that those are successful, at some point there could be systemic drugs?
Yes, so that would be the long-term goal, would be to try to give something systemic that would be able to get rid of senescent cells throughout your body. But I'll tell you, before we get to that point, we need to do a lot more research. Because I told you, there are good things that senescent cells do and bad things. And what we want to be able to do is target the bad cells and the bad secretions and preserve the good. And at the moment, we don't have enough knowledge to really say for sure that these are good candidate drugs for this kind of systemic administration. So we need a lot more research, but I am extremely optimistic that we're going to get there. There are new techniques in molecular biology that have given us enormous power to be able to identify small populations of cells and then target only the bad ones. So that's the big goal.
So I just want to reiterate how proud all of us who work at the Buck are of both Judy and of Unity Biotechnology. I mean, this is the first drug that's aimed at age-related disease that's in clinical trial. It's a big deal everywhere. It's a super big deal here, so I love to brag about it. Okay, so the research, as you mentioned, the research continues in your lab. Let's talk about some ongoing projects. One of the great things about being at the Buck is that you can collaborate with other people here. So let's talk about those. You're doing something with Simon Meloff. What's that about?
Yeah, we're doing a lot with Simon Meloff, actually. So Simon is a professor at the Buck Institute, and we're doing two things together that I think are really noteworthy. The first is, and they're both based on the idea that, actually we're doing three things together, the idea that senescent cells can drive these very different types of diseases. The first has to do with sarcopenia, the loss of muscle mass that is an inevitable consequence of aging. And we're being funded by a Japanese pharmaceutical company who has a big interest in sarcopenia. The idea is that there are certain stem cells in the muscle that undergo senescence, and they not only then fail to do their job, but they also start influencing the muscle themselves. And the idea would be to try to develop unique ways of getting rid of those senescent bad cells. The second thing we're doing has to do with cardiovascular disease. So there's this intimate relationship between your heart and your blood vessels. And both organs show an increase of senescent cells with age. And we know from mouse models and also from our cell culture experiments that those senescent heart cells and blood vessel cells can cause the neighboring cells to fail to function. So your blood vessels don't work as well, your heart doesn't work as well. What's unique about this program that Simon and I have put together is we've collaborated with a very talented translational professor at the University of Colorado in Boulder, who can obtain human blood vessels across the age spectrum, but also from people who exercise and people who don't exercise. And the idea is that we will be examining senescent cells from those blood vessels to, again, try to identify unique targets so we can get rid of the bad players. And the third thing we're doing with Simon is working on this incredibly powerful new technique of which he has mastered, which is to be able to identify all, or at least most, of the genes that are expressed in single cells, and to be able to do thousands of cells at a time. So we can identify small subpopulations that we think will be the best targets for, hopefully, a new wave of drugs.
And then you mentioned Julie Anderson. She's our resident expert in Parkinson's. Talk about senescence in the brain. What are you doing with Julie?
Yeah, so it's so interesting. Everyone knows that when the brain malfunctions in Alzheimer's disease and Parkinson's disease, it's the neurons that die. But it turns out that there's another cell type in your brain called the astrocyte. It's actually, there are more astrocytes in your brain than there are neurons. And the astrocytes are the cells that allow the neurons to function optimally. And so what we've shown with Julie is that in a model of Parkinson's disease, the astrocytes become senescent. They start secreting molecules that then impact the neurons and make the neurons fail to function. So this opens a new way of thinking about neurodegeneration, not trying so much to prevent the neurons from dying. That hasn't worked so well so far, but trying to get rid of those senescent astrocytes in the brain to allow the healthy astrocytes to take over and support the function of the neuron.
So now I want to get into this part of the seminar, and this is where you can participate. If you have questions, write them down on a card, raise your hand, and we'll get them up to me. So the question is, what do we do in the meantime? Unity's working on these drugs.
Yeah.
That's great, but what do we do now?
Yeah.
So I want to ask you the first question, which I know relates a little bit to your research. What do we know about exercise and senescence? Because we're very big on exercise here at the Buck Institute. If you've been around, you know that. So talk about exercise and senescence.
So when people ask me what is the single most important thing they can do to ensure a healthy life, I have to say it's exercise. It's true you want a good diet. It's true you don't want to smoke. It's true you want to choose your grandparents well, but we really want to exercise. And I'm not talking about running marathons. We're talking about moderate exercise. In fact, there was a recent study that was, in my mind, was just amazing. This was a study of obese people. See, these were all obese people, BMIs over 35. Those who were moderate exercisers, meaning 30, 40 minutes a day, maybe 3 times a week, and those who were sedentary. And the difference in lifespan was 8 to 10 years. So even if you want to eat and smoke, do your exercise. So what does it have to do with senescence? So this is a brand new field. It's something we're beginning to study. We do know that exercise can lower the amount of some of those molecules that senescent cells are secreted. We don't yet know if it kills off the cells, but it can definitely tune down all of those bad molecules that the senescent cells are secreting.
And I know it hasn't started yet, but you're hoping to do some collaborations with somebody at Sloan Kettering you were talking about. So what would that involve?
So one of the things that we are also interested in is accelerated aging. And there are genetic diseases that accelerate aging, but there are also treatments, good treatments, that result in accelerated aging. And one of them are many types of chemotherapies. So this has been known for years. There are, for example, thousands, hundreds of thousands of children who were treated for leukemia or lymphoma when they were children. And don't get me wrong, the chemotherapy cured their tumor. They were cured. But 20 years later, these children are now middle-aged adults, and by clinical standards, they look like they're 20 years older than they are. So they have increased cardiovascular disease, joint problems, cognitive problems, etc. So one of the things we know that these types of chemotherapies do is they induce senescence, and they induce a lifelong burden of senescence. And there's a group at Sloan Kettering who is now has some good preliminary data and is now embarking on a big trial to this is for breast cancer, so it's to put women who are treated with chemotherapy on an exercise regimen. And the preliminary results suggest that exercise can remarkably lower the burden of that accelerated aging that happens in so many patients when they receive the chemotherapy. So we're going to work with them to see what the role of senescence is in that exercise trial after the chemotherapy.
And before we have some great questions here, but I want to ask you, why is obesity such a problem with senescence?
Oh, inflammation, especially belly fat. It turns out that belly fat, the technical term is visceral fat, is a great depot for senescent cells. In fact, if you look in older people or animals, the organ that has the most senescent cells is usually this visceral fat, the belly fat. And of course, obesity is usually in the belly.
So I've got a number of questions people want to know about, and we talked about this beforehand. Anti-inflammatory diets. There's a lot of information. Eat an anti-inflammatory diet. What is that, and is there any preliminary evidence that it might impact senescence?
So it might. I mean, you also know that there are anti-inflammatory drugs, things like aspirin and ibuprofen. We've studied those drugs, and what I can tell you is that they are effective at reducing somewhat the secretions of senescent cells, which causes the inflammation, so that's why they're anti-inflammatory. But they're not totally effective, and they don't affect everything. They don't tune down all of the inflammatory components that senescent cells secrete. So I think those diets, they won't hurt. They certainly can't hurt, but we probably need something that is much more powerful, like a drug, like the drugs that Unity are developing.
So I got a lot of questions. People are really want to know if there are any supplements out there. If you take any supplements, what's out there that could possibly impact senescence, and if there's anything you want to say about the whole supplement field overall?
So again, supplements are pretty much like the anti-inflammatory diets and even the anti-inflammatory drugs. Some of them do have an effect on senescent cells. So one very famous one is quercetin. It's a natural product. You can buy it in CVS. And it's been shown to reduce some of the secretions of senescent cells.
So that's turmeric, right? The same thing as from turmeric?
Well, it's in turmeric. Actually, so the compound, the actual products like turmeric is actually a mixture of several compounds. Quercetin is one of the compounds that comes from turmeric.
Quercetin,
yes.
So everybody get that. If you need it written down afterwards, see me, I'll get you the information.
Yeah.
Okay, go ahead.
Yeah, so it works, but again, only partially. And so I think there's, again, very little harm. One thing I would caution is don't take megadoses of anything because everything in moderation, megadoses, we don't know what some of these compounds do, especially in pure form in megadoses, but it probably won't hurt.
I got a couple questions about stem cells and senescence, and you and I have talked before. One of the hot areas of research is people who are working with blood and trying to see if factors from young blood can help rejuvenate old blood, and I think senescence is involved in that. So what can you say about that new technology and.
Okay, and then we'll go to stem cells.
Yes.
Because they're different.
Okay,
They're
great.
separate.
Got it,
Okay.
yes.
So there are classic experiments. I think the best ones and classic ones were done by started in Tom Rando's lab at Stanford, and it's called heterochronic parabiosis. And what this means is that you surgically suture a young animal and an old animal together so that they share a blood supply. And the initial finding was that the young blood conferred something on the old animal that improved certain tissues, especially in the muscle. So if you injure the muscle, old muscle doesn't heal very well, but in the presence of young blood, old muscles healed much better. And that's true, and there are lots of people now studying what those factors are that the young blood can confer on the old. By the way, people don't normally do this surgical joining. So now they're doing just blood exchange. The animal committees like that much better. And the mice, I'm sure, like it much better. But so what we've learned, we're collaborating with one of Tom's postdocs, who's now an associate professor at the University of California in Berkeley. What we've done with her is we've shown that the opposite is also true. That is, old blood contains something that makes the young animal perform less well, makes those muscles heal less well. So our idea is that if we want to identify the factors that are in young blood that make old organs better, we're also going to have to identify what it is in the old blood that makes young organs worse and get rid of that. So we want to supplement and get rid of the bad stuff. And what we know that bad stuff can do, it induces senescence. So this is part of our collaboration, and so we're working on that to try to identify what those substances are and how to get rid of them.
So you wanted to talk about stem
cells, yes. So many tissues in your body have stem cells, at least those tissues that proliferate, and those stem cells are important throughout your life. They're going to help your tissue repair and regenerate. So what we know about senescence is, one, stem cells themselves can become senescent. And when they do, they can no longer proliferate. So they can no longer perform the rejuvenation and the repair. The second thing we learned is that nearby senescent cells, even if they're not a stem cell, the secretions of those senescent cells can make stem cells fail to function. They're just sitting here receiving these signals from the senescent cells, and they won't proliferate, and they won't go on to repair a tissue. So again, by eliminating senescent cells, we can release those good stem cells, and we think we can make them repair. Now here's another interesting thing. Most cancers contain some stem cells. They're cancer stem cells. They're mutant. They don't behave normally. But what we've been able to show is that senescent cells make factors that stimulate cancer stem cells while they're inhibiting normal stem cells. So we really want to get rid of those bad cells.
So talking about stem cells, and I got a question about this, there's a lot of controversy about all these clinics where you go and they insert stem cells in your knee or in your hip to try to, repair things. What do you think of those? And there's still senescent cells in those little joints, right?
That's the problem, is that unless you can clear the body of those factors that are inhibiting stem cell proliferation and function, you can put the cells there, but they're not going to function very well.
Let's talk about chemotherapy and senescent cells. So I know you've done research on that. For folks who might be unfortunate enough to have to undergo chemotherapy or have loved ones, what can you say about chemotherapy, senescence, and maybe some ways to deal with it?
So, as I mentioned earlier, most chemotherapies that are called genotoxic chemotherapies. So these are therapies that are designed to kill cancer cells by damaging their DNA. The problem is they also damage normal DNA, normal cell DNA, And those normal cells then are stressed, and many of them will become senescent. We now know from our mouse models, our transgenic mouse models, that if you can eliminate the senescent normal cells that occur as a consequence of the chemotherapy, you can prevent cancer metastases, at least certain cancer metastases. You can prevent the cardiotoxicity, the heart degradation that often limits how much chemotherapy a doctor can give. You can eliminate what has been called chemo fatigue. If you know anyone who has had chemotherapy, you know they feel terrible. We know if we can eliminate senescent cells right after the chemo, we can prevent that. And now we know that one of the things that chemotherapy tends to do is it tends to increase blood clotting, and that can increase the chance of a stroke. And we can show that senescent cells are responsible for that increased blood clotting. If we get rid of those senescent cells, we can restore normal clotting times.
So before those drugs are available, what can people do now?
Exercise. Exercise, yes, really, at least according to the Sloan Kettering folks. And those data look really promising.
Couple questions. Are there lab tests to measure low-grade chronic inflammation?
There are lab tests that measure low-grade chronic inflammation, but it's not at all clear how predictive they are. Some of them are for molecules that senescent cells produce. Some of them are for molecules that senescent cells, so far as we know, do not produce. One of the things we really lack in the field, and another big area for research, is developing biomarkers, that is, things that we can detect in a simple blood test, or even better, urine, or something where you don't even have to poke a hole in a person, that will be predictive, that will tell us what your biological age is and how you're responding to certain drugs. And by the way, Eric had on his slide that Birgit had just received a huge grant. This is part of our collaboration with Birgit, is to try to identify those biomarkers.
Got a question about autoimmune diseases like rheumatoid arthritis or Hashimoto's disease. So is senescence involved in autoimmune diseases?
Probably not. So rheumatoid arthritis is not the age-related osteoarthritis. Those, actually, they're usually more common in younger people, and they are a defect in the immune system. And so far as we know, senescent cells don't have much to do with those types of diseases, but I'm continually surprised by what people find. So stay tuned.
So this is a question I'm guessing from somebody who has some experience or familiarity with Parkinson's disease. So the plaques in Parkinson's disease come from alpha-synuclein?
They can.
They
yes,
can.
That's one of the.
So, and I know in Alzheimer's, they come from A-beta plaques and from tau. So does senescence have anything to do with those particular toxic molecules, and might at some point a systemic drug or a drug that can get into the brain, would that be helpful?
Well, one of the things we know is that when you have protein aggregation, whether it's alpha-synuclein, tau, or A-beta, or any protein, when you induce aggregation, which we can do these biochemical tricks to cause any cell to accumulate protein aggregates. That often puts the cell under enough stress that cells will undergo senescence. So again, one possibility is that those aggregated proteins are driving senescence, although probably the vulnerable cell is the astrocyte or the microglia rather than the neuron itself. Then, as a consequence of those senescent astrocytes and microglia, the neuron becomes extremely vulnerable, and eventually it dies. And let me say one other thing. In our transgenic mouse models, if we allow the neurons to die, then get rid of senescent cells, usually it's too late. So what we want is to be able to treat early, prior to after the neurons die. There is some stem cells in the brain that can be stimulated to replenish, but it's not very efficient. And so again, if senescent cells are playing a role in any of these neurodegenerative diseases, the key is going to be able to treat early, which means we need good biomarkers. We need to be able to identify people at risk.
Got a question about long-term low-dose use of Celebrex, and I want to add in things like ibuprofen and aspirin. If people take them over a long period of time, is it safe? What does the science show? And does it have a cumulative effect on senescence?
Well, as I said, these drugs do inhibit some of the secretions of senescent cells. They're not 100% effective. So far as I know, the epidemiological evidence for baby aspirin, so low-dose aspirin, is pretty encouraging. I can tell you right now, I'm a person that doesn't clot very well, and I did try taking baby aspirin for a short period of time, and I wound up with bruises all over my body, and I stopped. But I think, if you are prone to clotting, it probably won't hurt.
I got some questions about, I would call them stress reduction, so doing Qigong, listening to music, having a positive attitude. What do you think about that? It's probably not related, you can't relate it to senescence yet, but do you think there's a possibility? Lifestyle choices, less stress, could that help chronic inflammation?
Well, this is another big area that needs more research for sure. There are lots of anecdotal and semi-legitimate epidemiological studies that have tried to understand the effect of psychosocial stress on wellness and health span. We actually have now shared our transgenic mouse models with a lab in the University of Minnesota that has developed a very interesting mouse model of psychosocial stress. So no chemotherapy, no radiation. This is just pecking order stress. And so we will find out if senescent cells have any, so those mice are in general of poorer health, and they usually don't live as long as the unstressed animals. We'll find out if senescent cells have anything to do. He thinks it does. But anything you can do to be mellow, isn't that what California is all about? Isn't that why we're a superior state?
So, you're the pioneer in senescence, but I also want to acknowledge Judy for just knowing a whole lot about the whole aging field. In addition to the work that you and others are doing in senescence, is there anything in particular in the aging research field that you think is really promising that you're excited about?
One of the things I'm excited about is that traditionally, biomedical science would draw a line right about here, above the neck and below the neck. And in fact, the National Institute on Aging sort of funds projects that way. They have one that deals with the brain and one program that deals with the rest of the body. And I think what's happening now is that barrier between above the neck and below the neck are breaking down. And I think this is a really exciting area to understand how the brain affects the rest of your body and vice versa, how what's going on in the rest of your body can affect the brain. So I'll tell you of what I think is an amazing experiment. It wasn't done by me. I'm very jealous. I wish I had done this experiment. So these were cancer researchers, and what they were doing was giving mice two different types of cancer. The cancers would grow and kill the mice, no surprise. But then they broke the mice up into two groups. So one group is, we call our standard housing. We usually house our mice 4 to 5 in a cage about this big. Sometimes we give them little things to play with, but nothing. They really are pretty isolated and probably pretty bored. The other group of mice were put in what was called an enriched environment, huge cage, bells, whistles, lots of things to stimulate them. And those animals survived that cancer significantly longer because of this enriched environment. Now, this was a paper in a very well-known journal. You don't get away with just describing something. You have to get at the mechanism. And they did identify a hormone made in the brain that leaves the brain and can affect the rest of the body, and showed that that hormone was sufficient. Now they give that hormone to the guys in the little cage, and they survive the cancer much longer.
Wow.
So it's pretty interesting. And so this is something we're very interested in as well, as breaking down this idea that the brain is separate from the rest of your body. It's all, we're all together.
So I think there's some research happening at the Buck that involves leaky gut, right?
Yes. Yes.
And senescence. You want to talk, because
That's
everybody cares about leaky gut.
Yes, so Pankaj Kapahi's lab, who we're collaborating with also, he's been able, so he works in a model organism called the fruit fly. You've probably heard of him. The fancy name is Drosophila. Anybody who lives in Napa or Sonoma, you probably know these flies because they tend to love grapes. But these flies are amazing because the genetics is very well known. They can be manipulated genetically, and they're much cheaper to deal with than mice. So they have a gut that throughout life functions very similar to our gut, and as the flies age, like we do, the gut becomes leaky. And what happens there is two things. One is now the bacteria in the gut can leak out, and that can cause low-level inflammation, or even parts of the bacteria, not even the whole bacteria. And the other thing that happens is that the animals eventually can't digest food as well, and so they die. And what Pankaj's lab has been able to do is do a genetic screen and identify one gene that is very, very influential in how a leaky gut develops. And the beauty of that gene is that we humans have a very similar gene in our genome. So we're collaborating now with them to look at human cells in culture and mice to identify how that human gene might work to protect the gut during aging.
So we're almost at the end of this. I have saved the last question for myself. I have something in mind. But first, a question for you guys. How many of you here have kids or grandkids who are teenagers? Anybody deal with teenagers? I'm not going to ask you to raise your hand, but just in case those teenagers are really bright but maybe don't have a clear direction in life, this question is designed to give you comfort. So Judy, talk about your entree into science. When you were a young teenager, how did you get into science?
She knows the answer.
I do, it's true.
I went to an all-girls school through high school, and when it came time to go to college, I looked at the curriculum, and there was only one criterion that I cared about, and that was how many boys were in the curriculum. And that was science. Sign me up.
So the message is that great scientists come from all areas. I've interviewed scientists who say, I got a microscope when I was 8 years old and it changed everything, which it can, but if you have teenagers who maybe don't have quite the direction yet, take heart, because who knows how they might end up. So I want to thank you for coming. I want to thank Judy for being here.
Thank you.
She's going to hang around for a few minutes, so if you have questions, and I'll be here as well if you have any questions. Thanks for coming, and remember out there, donate. We can take credit cards today. Thanks so much for coming.